Hi, good afternoon, everybody. Everybody is here for virology, either 3310 or 4310, not French literature or the modern novel or any of that stuff, right? Okay. We were in the wrong room before, that's why I asked. Okay, so you should be registered for one of these two courses. Let me tell you a little bit about both of them before we start talking about viruses. So I'm Vincent Racaniello. I'm a professor up at the medical school campus. I've been up there for almost 30 years working on viruses, and that's my email address. If you have any questions anytime, just email. Also, and I also am on Twitter. Saul Silverstein, sitting in the back in the corner, raising his hand. Say hi to Saul. He is a He's a professor also, slightly older than I am. Also up at the medical school, has worked on viruses also for longer than I, and he will be giving some lectures. He'll be here almost every lecture, and so avail yourself of his expertise as well. And his email is here, sjs6. You can tell how old we are because we have really low numbers. VRR1, right? And sjs6. I don't know how that worked because you were before me. Yeah. Okay, and finally our TA for the course is Esther Francisco. Esther is my PhD student working on viruses. Okay, so avail yourself of our expertise. We're here to help you learn about virology. And that's her email address. So a few mechanics about the course. We are on CourseWorks, or I have a separate website because some people don't go to CourseWorks. So I have two different places, same material, mostly duplicated at both places. So CourseWorks is one, or this website, which is very long, so I've made I've done a URL shortening, bit.ly/cuvirology, bit.ly/cuvirology. Everybody, can you hear me in the back with all the noise? It's okay? Yeah, we have to close one of these because it's too loud. I'm sorry, yes. Thank you. So go to those websites to find out. I post all the slides ahead of time as a PDF, so you can take them with you, print them out, do whatever you want, take notes on them. And then I'm recording this. I'm recording the slides. I will put it up as a video cast for every lecture, and you'll find them there. So within a day or two after the lecture. So last year, about half the class showed up for every lecture, because if you can just hear me on a podcast, you don't have to come. And I think they did just as well as everyone else, so it's your option. I would love to see your smiling faces and have you ask great questions, but if you can't make it, that's fine. So on the websites, you'll have the schedule for all the lectures. There are readings for each lecture. There are textbook readings. The textbook has been ordered. It should be in the bookstore in a couple of days. There are assigned readings, and I will also post there the screencasts and the PDFs before the lecture, okay? And there's a separate CourseWorks page for graduate students and undergrads. Any grad students here taking the course? One? Okay. So the grad students also have a Friday session, right? And so the undergrads don't have to go to that. But for the grad students, they have to do more to get extra credit. And if there's only one student going, I'm not sure we'll do it, so we'll have to talk about that. Okay? Maybe they just didn't come today. All right, so all that is on the website, as well as the grading. We have 3 exams. We have a weekly online quiz to make sure you're keeping up, very short. And for graduate students, something more. No, she doesn't want to learn about viruses today. All right. The textbook is Principles of Virology, 3rd edition. I strongly recommend it. You may be able to get along without it. You could try it initially, but it will fill in all the gaps that I miss. It will have more than you need to know, but if you don't understand something and I can't explain it, and neither can Saul or Esther, perhaps the textbook will. But it's a great book, and it will keep you in virology for the rest of your lives. One of the goals of this course is to have you take virology with you through whatever you do in life, because it's important. I want you to understand all aspects of virology and not be confused as most people are, and this textbook will help you do that. I also want you to take a look periodically at my blog. I do a blog. I really believe in using social media to teach. Social media meaning interactive, where people can post on their own and they can comment. So I do a blog where I write periodically various things, and I also do a podcast called This Week in Virology, where me and a couple of my virology colleagues sit down and we talk about the important virology stories of the week. You don't have to do every podcast, you don't have to read every blog post. But what I want you to do is to get inside of virology. If you listen to podcasts and read the posts, you will see and think about what virologists are thinking about. And it will help you immerse you in the course and make it much more easier for you. If you just hear things about virology over and over, that will really enforce the learning. Okay, so those are all the materials we use for the course. Once again, the textbooks. Try and read the chapter ahead of time. I have a specific one associated with each lecture, the blog and the podcast. Okay. So this is an example of a blog post. I write about something that's interesting to me. It just pops into my head. I read a paper and I try and explain it. And this is written for the lay public. And you will find if you go here, I allow comments by anyone. It's a very interesting interaction with the public, because there are lots of very interesting diseases caused by viruses these days. The public is very engaged about it. Vaccination, for example, is a big issue with the public. And so a lot of people comment here, and you will see very interesting comments. This is the website for the podcast. Every week we have an episode with particular topics. In this one we covered 3 different stories. And again, I have a couple of my virology colleagues joining me. You can play it right here on the web page, or you can download it. So take advantage of those. Again, try and see how we are thinking about viruses, not just a lecture here where I'm talking at you, but casual conversations. And you can learn, I think, a lot more by listening in that way. Okay, that's the mechanics of the course. If you have any questions, come up afterwards. Office hours, by the way, will be on Wednesdays, which is today, right after this lecture in this room. Okay. We have viruses everywhere. We survive in a sea of viruses. We eat and breathe billions of them on a regular basis. For example, when you breathe, you're breathing about 6 liters of air a minute. It's a huge amount of air. You're sampling the air, which is full of viruses. We eat all kinds of food, of course, which is also contaminated. We touch all kinds of things. This doorknob, which may have been touched by thousands of people in the course of a week, is probably covered with viruses. And then we take our fingers, we put them in their eyes and our mouths, and we introduce viruses to us. Every milliliter of seawater has more than 1 million virus particles. Next time you go swimming, you take a mouthful of water. Just think of all the viruses that you're putting in your mouth. Even if you don't swallow that salty water, you're putting viruses in there. We carry viral genomes as part of our genetic material. Each and every one of you has in your genome the information to produce 1 or more viruses. Viruses also infect other things besides humans, of course. They infect our pets, the animals that we grow for food, pigs, cows, sheep, and goats. Wildlife, plants, insects, anything living has a virus that infects it. So they're everywhere. And viral infection can cross from one animal to another. There may be a virus of wild chimpanzees, and they can cross into humans. And that happens all the time. And these are called zoonotic infections, and we'll devote an entire lecture just to talking about those. Let's focus in on one type of virus, and I'll give you some numbers to emphasize just how staggering the numbers are. So this is a bacteriophage on the cover of the journal Cell. There are more than 10 to the 30th bacteriophage particles in all the waters of the world. This is an estimate made by sampling phages in different parts of the world. 10 to the 30th. This is a huge number. This is inconceivable, really. Virology? Yeah, it's too bad. It's his loss. So a bacteriophage weighs about a femtogram, 10 to the minus 15 grams. So if you multiply that by 10 to the 30th, it turns out that the biomass on Earth of bacterial viruses alone, so these are just viruses that infect bacteria, not us, exceeds the biomass of elephants by more than 1,000-fold. So that gives you an idea. Yeah, elephant is a big thing, right? By more than 1,000-fold. Now I know there are not a lot of elephants left, but still, that's a lot. And these are submicroscopic entities. So 10 to the 30th is huge. Here's another way to look at it. So the length of a head-to-tail line of 10 to the 30th phages is more than 200 million light years. And if you want, you can go to the website where this fellow made this calculation. That's the link to it there. How much is 200 million light years? That's another inconceivable number. Well, the closest galaxy to us, the Milky Way, is the Andromeda galaxy, which is 2.5 million light years away. So that's 100 times more. The phages would stretch into space 100 times further than the nearest galaxy. You can't even see this with the naked eye at night. So this is incredible. It's a lot of particles. Whales, that's a whale there. Whales are commonly infected by these tiny RNA viruses called caliciviruses. And whales, these viruses in people and in marine mammals can cause a variety of infections. In marine animals like whales and seals, dolphins, they can cause rashes and blisters and gastroenteritis. And they can infect humans. In fact, a very common way of them being transmitted is in these theme parks where they have the whales jumping out of the water and the people are sitting next to them and the whales are splashing them. And often the virus crosses over into people in those situations. So these viruses can infect humans. Infected whales secrete more than 10 to the 13th caliciviruses a day. Okay? Each whale every day is secreting this into the ocean. So this is adding to the biomass of viruses in the ocean. It's a huge number. So one of the things I want you to get from all of this is that when we talk about viruses, the numbers are astronomical. They're bigger than anything else that we know about on Earth. And in the course of this course, we will try and understand why viruses produce such large numbers of progeny. There are 10 to the 16th HIV genomes on the planet right now. It's another big number. And this is, of course, because millions and millions of people are infected, and each infected person makes a huge number of particles every day. With this number of genomes, it's possible that there are already genomes that exist that are resistant to all the drugs we have. We have over 20 of them, or that we'll ever discover. If we keep making drugs for years and years and years, somewhere out there, there's already a resistant virus. And this is one of the reasons why we have to use combination drug therapy to treat a virus like HIV. And we'll talk about this in some detail. Despite all of this, despite the viruses on the doorknob and floating around in this room, we're pretty healthy most of the time. And that's because we have a great defense system that has evolved along with viruses to fight infections. Viruses have been with us since the beginning of life, maybe even before, some people believe. And so life has evolved along with viruses infecting them. So they have, in order to survive, they have to evolve defense mechanisms. So we have great immune systems that keep us healthy most of the time. But if our immune system is down, if it's defective or suppressed, so if you get an organ transplant and you receive drugs to immunosuppress you so you won't reject the transplant, your immune system is dampened. Or if you have AIDS, in which case you're also immunosuppressed because the virus is killing the cells of your immune system, then you have problems with common virus infections and bacterial and fungal as well, that would normally be nothing or benign to us, but is lethal in an immunosuppressed person. So how infected are we exactly? A couple of slides to emphasize this. Every one of you is probably infected with at least 3 of the 9. I think you said that last year, right? And let's see which 3 we have. These are the 9 known herpes viruses: simplex 1 and 2, VZV, cytomegalovirus, Epstein-Barr, HHV 6, 7, and 8, and B. So which 3 would everyone have, Saul?
1, VZV, and EBV.
Okay, HSV1, VZV, and EBV. 3 of the 9 known herpes viruses. The B virus is 100% lethal, so none of you are infected with that. And once infected with any of these, you are infected for life. And every one of your cells is infected with viruses. You have thousands of copies of retrovirus genomes integrated into your DNA. About 8% of your DNA, your genomic DNA, is made up of these retrovirus sequences. Now in us, they happen to be defective. They have mutations in them, so they don't produce virus particles. But in other animals, these endogenous retroviruses, as we call them, do produce infectious virions. You will pass these retrovirus sequences on to your children, and they will do the same on and on and on. And this has probably been going on as long as there have been Homo sapiens, and probably the hominids before that. And it's an interesting question. Why do we have 8% of our genome as retroviral sequences? Maybe they're doing something for us that we don't know about. So we'll talk about that as well in another lecture. What are the genomes doing there? Do they help us at all? They're also a reservoir for viruses that have set up residence in your lungs and your GI tract. Your lungs and GI tract are constantly sampling the environment, so you have lots of viruses in them, a few other places as well. We're all colonized by adenoviruses, coronaviruses, rhinoviruses. These are common viruses that cause respiratory infections. As you know, we actually have more bacteria in us than human cells, and a main place where that is in the gut. And these are good for us. The particular blend of bacteria that we each have in our guts is important for our well-being. And they have their own viruses as well. And we're just beginning to study those and understand the beneficial role of these viruses for those bacteria. They've been with us, as I said, since we've evolved, and probably before that as well. Now, this is a slide I just stuck in this morning because we heard a lecture from a virologist today by the name of Herbert Virgin. And he showed this slide, which is the estimated burden of chronic viral infections in humans. Now, chronic viral infection is one that goes on for years and years. We'll talk about the different kinds of infections later on. But these viral infections go on for years and years. And this is a graph showing on the y-axis the estimated number of infected individuals in the millions. So we have 10 to the 0 up to 10 to the 4th million. So the population of the world is almost 7 billion people or so, which would be somewhere in here. So you can see some of these viruses infect everyone. The endogenous retroviruses are in everyone, so that's the world's population there. And then we have anelloviruses, which are curious DNA viruses. No one has any clue what they are doing, if they cause any disease, but most people seem to be infected with them. And here are our herpes viruses. Look, HHV7 and 8, VZV, EBV, CMV, all infecting large proportions of the population. Other herpes viruses as well. And then we go down in prevalence as we reach HTLV1, human T-cell leukemia virus 1, hepatitis delta virus, HIV, and some in between. So the burden of chronic viral infections on their own is huge. And we think not all of these are bad. I don't want you to end up at the end of this course thinking that all viruses are bad. Some of them are probably good. In fact, many of them are probably good for us in a variety of ways. And hopefully we get to talk about that as well. So I think viruses are amazing. You probably have some fascination with them, or else you wouldn't be here. And this course is going to tell you why I think they're amazing and why they're amazing. How do they get to be amazing, and how do they work? So I want you to see the big picture in this course. I don't want to teach you 50 different viruses and how they replicate and how they cause disease. That's not what this course is about. That would be a survey course, and I don't believe in that kind of teaching, not at this level. I want you to see the big picture, the overview, so you can look at any given virus and figure out how it replicates and how it causes disease. So I want you to learn general things about replication, spread, and disease causing. I want you to think about, I'll show you how to think about virology as an integrated discipline. And again, not a collection of viruses, diseases, genes, but the whole picture. And this will be clear to you by the end, I think, if it is not now. I also want you to appreciate what viruses do to pervade the ecosystem to the extent that we have just talked about today, the numbers that you see. And then you'll also learn the fundamentals about viruses. And these are things that most people don't understand, and that is why they are frightened of viruses. The unknown is the most frightening thing, and I'm going to help you understand viruses so you'll be less frightened about them. Now, virology requires that you know a little bit about a lot of subjects in biology. Viruses use cells in order to replicate, so you have to know how cells work. For example, does everyone know the difference between translation and transcription? Everyone knows? Because last year, not everyone knew. So I'm glad that you guys do. Maybe you got a better background. So I'm going to be saying translation, transcription, nuclear import. And if you don't know what it means, you better either find out or ask me, because you're going to get lost. Because if you think when I say translation that I mean RNA synthesis, that will be a problem. It's going to be confusing. And virology will always test your ability to put things together. You're going to have to do that. You're going to have to integrate your knowledge about how a cell works in order to figure out how a virus genome replicates, for example. Now, you have to know certain facts, and at different points in this course, I'll tell you the things you must memorize. Not everything. You're going to have to memorize certain things. But you're never going to be able to memorize all the combinations of facts that tell you how viruses work. You're going to have to think. Okay, so memorization is not enough. I want you to actively think and say, does this make sense from what I know? And of course, there are details to learn about different viruses. We will point those out, and that is some of the fun part about learning about how viruses work. Now, here's a very nice quote from a writer that I happen to like from Wired magazine in 2008. And I think this may summarize the philosophy in us teaching this course. One of the reasons kids get bored by science is that too many teachers present it as a fusty collection of facts for memorization. This is precisely wrong. Science isn't about facts, it's about the quest for facts. The scientific method, the process by which we hash through confusing thickets of ignorance. It's dynamic, argumentative, collaborative, competitive, filled with flashes of crazy excitement and hours of drudge work, and driven by ego, our desire to be the one who figures it out, at least for now. So as we go along, we will try and tell you how we found these things, these facts that we are telling you about viruses. Because I think that is really the basis for science, not the fact itself, but how we got to that point. And this is a very important point here, which I can't overemphasize. We all like to think that we do science to figure out things to make life better for people. And it certainly is a component. But ego plays a huge role. Ego meaning I want to be the first to figure this out. And if that drives science, that's fine. There happens to be some untidy side effects of having big egos in science. But it does drive science. The desire to be the best and to be the first is the main driver of progress in science. Viruses infect every living thing, as I have said, but most people think that viruses are bad news wrapped in a bit of protein. I forgot who said this. A scientist said this not too long ago, maybe many years ago. They're not all bad news. Many of them are. And in fact, we are biased because scientists, virologists, study the viruses that make you sick because you want to figure out how to prevent you from getting sick. But there are many viruses out there that don't make you sick and that are not bad news at all. And we'll try and emphasize those in this course as well. Viruses do much more than cause disease. So the big picture I want you to take away from this course is that all viruses follow a simple three-part strategy to survive. They all package their genomes inside a particle used for transmission from host to host. So there's a genome and a particle. And the particle protects the genome as it goes from one host to another. The genome has all the information you need to make new viruses. We'll go through this infectious cycle in this course and explain how it works for different genome types. And then finally, all the genomes can establish themselves in a host population so that they persist. Influenza viruses have been with us for hundreds and hundreds, perhaps thousands of years. That's because the virus has evolved a strategy to do that. If it didn't, it would be gone. So the genome encodes all that information, the ability to stay within a population, and when the virus encounters a new host, goes from a human to an animal, or vice versa, the genome can be plastic. It can change to adapt to the new host. So if that's it, then why study virology? Why take this course? Well, the tactics to achieve that three-part strategy are very diverse, and I want to tell you about them in this course. There are many different particles out there with countless diversity. The size and the nature of genomes are different. There are odd particles, and there are very interesting coding strategies. So the three-part strategy holds, but the way that viruses do it are very diverse, and I want you to learn about that, and we'll try and make sense of it to unify it in a way that you can understand it. There are two simple facts, though, that we can use to simplify this enormous diversity. And the first is that all viral genomes are molecular parasites. That is, they can only function after they get into a cell. A virus cannot do anything on its own. The virus is sitting on these chairs or floating in the air will do so forever until they degrade and break apart, unless they encounter a host and cells within that host in which they can multiply. So they are parasites. They are molecular parasites. And we'll see in this course the extent to which viruses are parasitic. In different kinds of viruses have different levels of parasitism. Some completely depend on the host for everything. Some viruses have only one or two genes, whereas many viruses are huge and can code hundreds of genes and are much less dependent on the host. But all of them are parasites. At least the ones that we know now are parasites. And the second very important unifying principle is that all viruses have to make messenger RNA. And that messenger RNA has to be able to be translated by host ribosomes. So no matter what the virus, if you just think of this one fact throughout this course, it will help you enormously. Every virus has to make mRNA at some point. No matter what else goes on, it has to make mRNA, and it has to be translated by the host. So they are also parasites of the host protein synthesis machinery. We don't know of any virus that encodes its own complete translation machinery. And that, of course, includes ribosomes, tRNAs, all the amino acids, the synthetases, and so forth that you need to make proteins from an RNA template. No virus can do that on its own. So they're obligately devoted to using the host translation system. Now, because of this, because of the parasitic nature of viruses, when you study them, you by definition learn about the host as well. So if you understand how a virus parasitizes the host translation system, how it utilizes it, you will learn about the host. And viruses, in fact, have turned out to be wonderful probes for understanding host functions. Now, a word of warning. Be careful about anthropomorphic characterizations of viruses. What do I mean by that? Viruses don't think. Well, you say obviously they don't have brains, but people often say a virus will employ this strategy or ensure its survival, exhibit, display. There are many, many words. In fact, there's a little bit of text in chapter 1 that deals with all the words that we don't like to use. Because the point is, and it's not just an exercise, it gets you to thinking about viruses as passive vehicles. Viruses have evolved to undergo chemical reactions which propagate them. They don't want to do something. They don't want to kill a host. They don't want to make a lot of particles. It's just a chemical reaction. And I think if you avoid using human-like words to characterize what viruses do, you set yourself in a better frame of mind for understanding how viral replication cycles occur. It's very hard to do. I'll slip all the time in talking to you. And in fact, you can probably find in our textbook all the mistakes we made where we left viruses doing something in. But I really think it's a good exercise to undertake to get you thinking about how viruses work in the correct way so they don't achieve their goals in a human-centered manner. And here's the key. They survive because they make huge numbers of mutants at every replication cycle. Those big numbers that I showed you earlier, they're all a consequence of making a lot of particles every replication cycle, more than any other form of existence on Earth. They make more of themselves, more progeny in a given time than anything else. And that's why they're successful, because a lot of those particles are no good. They don't work under certain conditions. The ill-adapted are gone. They never replicate. And if a condition changes in an animal or a host species, there are viruses there that can take advantage of that because there are so many of them, because there are millions and millions of particles produced in every cycle. There's always one that can take advantage of a different condition, a different animal walking by, a different weather, ultraviolet rays, you name it. It's really a beautiful example of selection of the fittest, and they're great Darwinian machines. I think there's no better model for the concept of survival of the fittest. So if Darwin knew about viruses, that's what he would have studied, because it's a lot easier than cataloging birds on a remote island. He can grow billions of viruses in a day and watch evolution occur in your cell cultures. So let's think about this a little bit more. Viruses, of course, depend on their host for survival. If there weren't any hosts on Earth, or if there weren't life, there wouldn't be any viruses. So what does that mean? If the virus is too successful, if it kills their host, then it doesn't have a host to grow in, right? On the other hand, if viruses are too passive and the host defenses overcome an infection, for example, the virus can be eliminated as well. So it's a little paradoxical, right? You have to have a balance between killing and existing with the host. And this has been debated by many, many people for years. It's a common misconception that viruses evolved to get more virulent for humans. So I saw in the past year in almost every newspaper the notion that this influenza pandemic strain was going to evolve to get more virulent in people. Why would it do that? It makes no sense whatsoever. Why would it kill and debilitate the host and affect its spread? Now, I'm giving a little anthropomorphic characterization here. I don't mean to. But in terms of selection and propagation, it makes more sense for the virus to coexist peacefully with the host. There are examples of virus infections that are terribly lethal, but they, for the most part, don't spread very much. Yes?
Right now, do they think the bacteria was before the viruses, or where do they place them?
That depends who you ask. You'll get both answers. Some people think, well, first there was a soup of, before any life, there was an organic soup from which life evolved. Some people think viruses could actually replicate in that soup before there were cells. So that's one, because theoretically it's possible. You could have just a simple nucleic acid genome multiplying in an organic soup. Others think that cellular life came first. It's a big debate because we don't have fossils like we do for a lot of other evolutionary questions. But it's a lot of fun to talk about. We're going to talk about evolution, and we'll do that. So how do you balance this? Survival of the host and survival of the virus. This is a really important underlying question. You don't want to kill the hosts off because then you have no host to multiply in, but you can't be wimpy, otherwise you're not going to get anywhere as well. So there's an incredible balance, and this is one of the big questions that virologists try and understand. And it's very difficult to study in people, so we turn to other models for doing that. And all of the twists and turns that allow viruses to propagate, in, whether it be in a cell or a tissue or in an entire organism or in the whole population, this provides insight into mechanisms that we couldn't study any other way. And that's another theme in this course. We can study aspects of life that you can't do without virus infections. So when this course is over, not only it's going to be spring, which is really nice, you will have significantly better insight into how cells work and how they interact, not just viruses. Because viruses are wedges or hooks. They target processes to their advantage. So when I explain how a virus replicates, you're going to better understand the cellular process. You're going to have a better knowledge of the way information is stored and decoded in viral genomes. And this is far more diverse and complicated than occurs in our cells, for example. Many, many different strategies. We have very tiny genomes. How can you pack so much information into a tiny viral genome and have it work? We also will have a basic understanding of viral pathogenesis, which is the process by which viruses cause disease and infectious disease in general. You'll be able to better understand what's happening to you when you get sick from a viral infection. And when your family says to you, should I be vaccinated, you'll be able to make a better decision about that as well. So why do you feel badly when you get a viral infection? Immunology will explain that. And that's one of the things you'll be able to do when the course is over. In addition, you'll be able to defend these assertions. Well, you may disagree with me, which is fine, but I think you'll see by the end of the course that viruses have more biological diversity than all the rest of life. Well, not life, because I don't think they're living, but let's just say that bacterial, plant, and animal kingdoms put together. Far greater biological diversity. We are actually not at the top of the food chain. I think viruses are. I think the tiny may well inherit the earth, although they can't get rid of us, otherwise they wouldn't be able to propagate. And despite being very small, most viruses, it turns out, have less than 15 genes. We have tens of thousands of genes in us. Some viruses only have one gene, and that's enough for them to propagate and be very successful. They excel at survival in what is a pretty harsh world, and the molecular biology is very sophisticated. And finally, virus-host interactions define interactions of fundamental and practical significance that we really need to understand if we want to protect against serious virus infections. We have very, very rudimentary knowledge of how viruses and hosts interact, but we're getting better at it, and I hope to tell you some of the results that we've had. Finally, I want you to come out of this course and be able to look at a news report like this terrible one from CNN, which focuses on all the sensationalism of swine flu. You'll be able to read newspaper articles, online articles, and blogs and websites, and you'll say, this is nonsense. This can't be, it's just sensationalism. And remember that newspapers and TV programs have to be sensational to make money. For some reason, that's what people respond to. So here we have a CNN headline from actually one of my readers on the blog sent this to me last year. This was from last summer, I believe. A paper came out in the New England Journal of Medicine where they had infected ferrets with the new pandemic influenza virus. And so they put this headline up without telling the listeners that this referred to experiments in a ferret. And it can ravage the lungs, spread through respiratory systems, causes lesions, doesn't stay in the head like seasonal flu. I don't know what that means, because flu typically is a lower respiratory tract infection. The last point, survivors of 1918 flu are immune. That's true, because the virus is quite related antigenically to that, and we'll talk about that. But an animal model is great for understanding aspects of virology, but it turns out that the virus was quite benign in people for the most part. And so the animal results did not predict what was happening in people. But it was just a splashy, sensationalist headline, and this is what we're trying to combat. So I want you to be able to look beyond this, look through this kind of sensationalism. So we're going to talk mainly about animal viruses in this course, but we will talk about some viruses that infect bacteria because they are great model systems. Not a lot, but enough to explain certain principles. And many viruses we'll not pay any attention to, sadly, because of time constraints. But once you take this course, you'll be in love with virology and you'll be following viruses for the rest of your lives. So I'm not worried about that. So let's talk a little bit about viral origins and history. How old are viruses? Well, with a few exceptions, and we'll talk about the exceptions, there's no fossil record. So it's very difficult to know how old they are. You can get estimates for how old they are by looking at the sequence change. So viruses change in their sequence a certain amount each year. It's called the molecular clock. And you can back calculate using a molecular clock calculation. And for some viruses, it places them among the dinosaurs, 150 million years ago. And this is all possible now because we can sequence viral genomes, and there's a whole new area of bioinformatics that uses mathematical algorithms to do these calculations. In theory, as I mentioned earlier, viruses could predate cellular life. They could have existed in a soup. We'll talk a little bit about those early origins, but my feeling is that probably viruses were around even before cellular life evolved, but I think it will be hard to prove. We do have lots of early records of virus infections in prehistory. All right, so let me tell you a few of those. So there are ancient references to viral diseases. So here we have from 700 BC from the Iliad. And it says, here this firebrand rabid Hector leads the charge. So rabid, referring to this virus disease passed on to humans by rabid animals. And they didn't know it was a virus at the time, but it meant that viruses were around. And here's another one from about 1500 to 1300 BC. It's an Egyptian carving, which shows this young priest with this leg, it's called dropped foot, and a withered calf, which is very typical of polio. So we think this is the first recorded characterization of poliomyelitis, which is also caused by a virus. So the historical records are full of references to diseases which, when we look at them now, look very much like either viral or bacterial diseases. And there are entire books written on this subject which discuss, for example, how battles were lost due to infectious diseases. Now, of course, no one knew what these were, but clearly the virus infections were around as long as people were around. You can also understand from looking at the way people prevented virus infection. So as long ago as the 11th century, there were practices in place to prevent virus infections. Again, it wasn't known that they were virus infections without knowledge of the agent. And a very famous practice was called variolation. And smallpox is a disease that we can very clearly see in historical records because it has very prominent symptoms. It has a very prominent rash, it's fatal, and so we can track it historically. What was noticed very early on was that once you got smallpox, if you weren't killed by the disease, you survived and you never got it again. So people were smart. They said, I wonder if I naturally infect myself or give myself this disease, can I protect myself? So the practice of variolation arose where they would take the pustules from a smallpox victim, and these are full of virus. They would grind them up and inject them into someone else. And if you didn't die from that, because you did often, you were protected against the disease. And this is actually something brought to the UK, England, in the 1700s by Lady Montagu, who was the wife of the Turkish ambassador. And she went to Turkey and found this practice being done, where you would go to a woman who had a little room, and she would have a vial full of ground up crusts from smallpox victims. And you would give her a vein, and she would shove some into it. And as I said, that has a lot of virus in it, and if you didn't die, you would be protected. So these are the first examples of immunization. But it wasn't until the later 1700s that Edward Jenner established true vaccination. Jenner realized you can't infect people with the agent that causes the disease. He didn't know what the agent was, but he knew there were forms of the disease that were milder, so his idea was to use that. And when we talk about vaccines, we'll go through that. Now, the concept of microorganisms is not developed yet at this point in history. And really the first beginnings were from the Dutch lens maker, Leeuwenhoek. He made lenses for glasses and all sorts of telescopes, and he made a microscope one day. You've probably seen images of this very crude microscope, and he started looking at things under it. And he found these wee animalcules, things moving around, microbes, bacteria, yeasts, fungi. And from that work, initially, became the acceptance of the idea that there are things smaller than us, microorganisms. And it took Pasteur to show in the 1800s that these are generated by reproduction. They don't spontaneously reproduce. There was a whole train of non-scientific thought where people thought that these wee animalcules, these microbes, arose by spontaneous generation. Huge debates between Pasteur and his opponents. But he finally prevailed and showed that these reproduced, not by spontaneous generation. But the germ theory of disease, which links a microbe to a disease in humans, wasn't formally established until Robert Koch did so in Germany in 1879. He said, we have microorganisms and some of them can cause disease, and he proved it. But he didn't know anything about viruses yet. These are all bacteria that we know now. Now, this is how Pasteur got around the problem of spontaneous generation. So he took a flask with broth in it in which microorganisms could grow, and he boiled it to sterilize it. And nothing would grow in it unless he put air into the neck. So any contaminating microorganisms from the environment would get in here and they would be stuck down here. They would never make it up here. And these flasks actually still exist at the Pasteur Museum in Paris. They're still not contaminated. So we did a podcast about this. There's a professor at Columbia who's a historian of science, and he talked about this in our podcast number 35. So that was the proof that microorganisms reproduced. Viruses came around about the end of the 1800s. The first virus to be discovered was one that infects plants, and this will be a theme recurring. You'll see many discoveries in virology occurred using plant viruses. This is a tobacco leaf, and it's infected with a virus that causes these mottled spots on it, and eventually this leaf will wither up and die. And if you want to grow tobacco, this is very bad, for cigars and cigarettes and so forth. So the scientists at the end of the 1800s was trying to figure out what caused this. And back in those days, the standard method which Pasteur had developed would be to take what you suspect to be a microorganism and filter out the liquid, take the concentrated microorganism, and in this case rub it on the leaf. Well, this fellow did this over and over, and what he found was that what caused the infection of the leaf was actually going through the filter. So this is what these filters look like. These are called porcelain filters. You basically had a vacuum flask, and then you have this chamber up here. You could pour in liquid up here. That's what the yellow material is. And this is the porcelain filter. And had very fine holes in it which would stop bacteria from going through. So they learned this empirically by making porcelain with different sized pores. And so the bacteria would remain here, and what went through was sterile. And what was found with tobacco mosaic disease was that the agent of the disease was down here. It wasn't hung up by the filter. And this was a fellow named Ivanovsky who did this in 1892. He found that the agent of the disease, tobacco mosaic disease, passes through the filter. Okay, so you filter it, you take this liquid, you put it on a leaf, the leaf gets infected. And that didn't make sense to anyone because up to that point everyone understood that microorganisms were retained by these filters. So Ivanovsky said, well, it's a really small microbe, and he let it go. We cite that as the beginning of virology, but he really didn't understand that he was working with viruses. It was about 6 years later, Bayerink made the same finding. The filtrate is what causes infection of the leaves, but he said, this is a distinct pathogen. This is just not just a microbe that we've been studying so far. This is something new. He didn't know what it was, but he made the conceptual leap that this is a new kind of agent, and that's really the beginning of virology. In 1898, the first animal virus was discovered, the agent of foot and mouth disease, which is a disease of cattle. And the key concept here is not only that these agents are small, but they need a host to replicate. They don't grow in broth. So Pasteur's microorganisms would grow in broth. But if you took these filtrates of tobacco leaves, they wouldn't grow in any kind of broth. You had to put them on a cell. And the same with the foot and mouth disease agent. You had to put it in an animal. There were no cell cultures back then, and it would only grow in the animal. So very small and needs a host to replicate, and from that arose the concept of virus. And back then, they used these porcelain filters which had a pore size of about 0.2 microns. All right, a micrometer is 1/millionth of a meter. This is really small. And this held as a definition of a virus until maybe 5 years ago. As we'll see, now there are some viruses that are bigger, so we're not quite sure where to put them anymore. But 0.2 micron filters were the defining agent for the size of viruses for many years. So here's some of the years of virus discovery. 1901, the first human virus, yellow fever. 1903, rabies, variola, smallpox. 1906, leukemia virus and polio in 1908, and a sarcoma virus in 1911. Bacteriophages were discovered in 1915, and then flu not till 1933. And then shortly after the isolation of influenza virus, in the 1940s, the first vaccines against those viruses were made. So virus means toxin or poison in Latin. That's where the name comes from. And I will use the term many times in this course, probably every lecture, virion. So we'll say virus is a generic term when we refer to viruses. We're just talking about the general concept of a virus. But a virion is actually the infectious particle. Okay, so remember that. Virion, whenever I say virion, it means a specific thing. And the particle has to be infectious. It's not a non-infectious particle. That's a non-infectious particle. A virion is the infectious particle. So here are some examples. These are electron micrographs. In the 1940s, the electron micrograph was developed, microscope was developed. We got the ability to see what these things were that were passing through 0.2 micron filters. And up on the upper left is a bacteriophage. This is one of the funkier looking ones. It has tails. You've seen these pictures before. A neck and a head where the nucleic acid is. Here is tobacco mosaic virus. It is a long rod and the nucleic acid is wrapped up in it. This is a rhabdovirus like rabies. They're bullet-shaped virions. And gee, I don't know what this virus is. I forgot. A rotavirus, for example, a non-enveloped virus. So when people began to look under the microscope, they said, wow, these are discrete particles associated with this infectivity. We know many details about viruses. Let me shut the light off for this one. This is poliovirus, and since the 1980s, late 1970s actually, we can figure out the three-dimensional structure of viruses. I'm sorry, this should be in white because it tells you the chemical formula for this virus, which is poliovirus. You can see it has 332,652 carbon atoms and hydrogen, nitrogen, oxygen, phosphorus, and sulfur. So we know the exact chemical composition because we know the sequence of all of the proteins of this virus. But actually that chemical formula isn't correct. It's just an average because as you'll see, every virus in a solution is different. It has a different genome sequence. So the precise composition of phosphorus and carbon and so forth is different from each virus, one to another. So this brings us to the question, what is a virus? I've been telling you some history and what we're going to learn, but what is it? What's the definition? Well, it's a very small infectious, obligate, intracellular parasite. And this definition has held for years. The very small is kind of problematic now because there are some pretty big viruses, as you will see, that you can actually see in a light microscope. But anyway, we'll use this as a working definition. A parasite, of course, is an organism that benefits at the expense of a host, usually a different organism. So viruses are parasites, and they're obligate intracellular parasites. They have to go inside a cell. You know, there are many kinds of parasites that live in your intestines, worms of various sorts for intestines. They are not within your cells. Viruses have to reproduce within your cells. Intracellular parasites. Now, I do not think that virus particles are living. I think I shut it off. Okay. I do not think that virus particles are living. It's objecting to my saying that to you. Many people debate this. But the particle, not virus, the particle, the virion, I should actually have virions are not living. A virion is just a chemical. It's not living and has a potential to live when it goes into a cell. So maybe you could view the infected cell as living, but the particle itself is not living. But this is debated, and that's just my view, and you can have your own as well. I think they're chemicals and cannot reproduce on their own. You need a cellular host in order to reproduce. So on my blog, I put a poll about a year ago. Are viruses alive? You should go take this poll at some point when you feel confident in your answer. Here's the link. I had the options were yes, no, or they are something in between, or I don't know. Now, I shouldn't have put the something in between because too many people picked it. But we have 1,500 responses, 29%, 32, 33. So it's pretty evenly divided among the three. So I don't know who's answering this. I think it's all kinds of people. I think there's some virologists here as well as non-scientists as well. But when you feel you can answer this question, or maybe you already have an opinion, go there and take the survey. Now, viruses are very small, as I said. Here's a picture that gives you some idea of that. So here's a carbon atom up here. It's obviously magnified a lot. And these are all various structures at the right scale for carbon. So here's a tRNA, an antibody, myosin, actin. And here's a ribosome and poliovirus. So you can see polio is about the size of a ribosome, considerably bigger than carbon. Now, we take this whole panel and we shrink it so it's this size, and now we have some other viruses that are really big, and so that's why we have to shrink these to go down. We have HIV-1 up here, which is quite large, tobacco mosaic virus, and then a bacteriophage, and here's E. coli. So E. coli is huge. And these viruses are quite big. But we have another virus, which I'll talk about in a moment, which is slightly bigger than E. coli, at least in diameter. So this gives you some relative ideas about the size of viruses. Now here is a cell with some viruses near it to give you more of an idea. So here's a eukaryotic cell, right? You got your nucleus here, endoplasmic reticulum, Golgi, mitochondria. I'm helping my kids study biology, so I know all this stuff. You know. And we have some virus particles here at the surface. And here's a magnification. The big guy is herpes virus, and this is poliovirus. So herpes is quite large compared to the cell. Of course, this is just a two-dimensional slice. The cell has volume, of course. It's going to be much bigger than the virus, but it gives you some kind of idea of the size of viruses compared to cells. How many viruses can fit on the head of a pin? Well, it depends on the virus, of course, but if it's a rhinovirus, one of the causes of the common cold, you could put 500 million on the head of a pin. But the bigger viruses, many fewer. When you sneeze, you fire an aerosol that contains enough viruses to infect thousands, and we'll talk about that as well. Very small. What does that mean? Well, Mimi virus was discovered, I don't know, about 5 years ago. The name means microbial mimic. Mimi virus was isolated from a water tower in France, and the individuals who isolated it thought it was, they looked at it under the microscope, they thought it was a bacterium, so they put it in the freezer, and it sat there for a couple of years till they went back to it, and they realized it was a virus. It was just really big. It grows in amoebae in the wild, and it's about 750 nanometers, or 0.75 microns in diameter. So remember, the filter we use to filter out viruses is 0.2 microns. So this will not go through the filter. So if you use that kind of filter to look for viruses, you will miss the Mimi viruses. So you can't use really 0.2 micron as a definition for viruses anymore because we have these big ones. The DNA, this is a double-stranded DNA virus, is huge, 1.1 million base pairs with 1,262 open reading frames or potential proteins. This is huge. Right? Some viruses only encode one protein. And it encodes many components of the translation system. tRNA synthetases, translation proteins, tRNAs. It even has components of DNA repair pathways. It's got a lot of genes. So this is getting to be a blurring virus between real viruses and host cells. And what the interesting question is now, are we going to find even bigger viruses that run the continuum from the smallest virus all the way up to the host cell? Will we find a virus that has almost everything it needs to replicate? We'll see. Challenges the frontier between viruses and cells. So this is a cell infected with Mimi viruses. So this is similar to the cell I showed you before. It's a nucleus. Actually, that's not the nucleus. Right here is the nucleus. And these are Mimi virus particles, MV. You can see them. This is actually an electron micrograph, but you could see these in a infected cell in a light microscope. If you want to know more about these Mimi viruses, there's a website called giantvirus.org. So viral attributes. What is a virus? Continuing this discussion, the genome is either DNA or RNA. So our genomes are DNA, but viruses can be either one. In the host cell, the viral genome directs the synthesis of new particles by cellular systems. New particles are made by assembly from parts. The viruses make the parts, and they're put together. And the progeny particles transmit the infection to the next host or organism, and then the infectious cycle begins again. So these are, again, properties of a virus: small, obligated intracellular parasites that have to go through these steps to replicate. When a virus infects a cell, it needs the cell, of course, so a viral infection is an exercise in cell biology. These are some of the functions of the cell that are needed for viral propagation. You need translation, of course. You need energy. Viruses can't make energy, can't make ATP. They don't do respiration, or what's the other one? Fermentation. Nope, can't do that. They need enzymes for replication and assembly, and transport pathways. They have to move stuff around the cell. They can't do it, so they use cellular transport pathways. So when you study virus replication, you're going to study all of these things that go on in cells. And this is a really important concept that we'll come back to later. Viruses replicate by assembling components. It's like an assembly line. You make the parts, you put them together, you get a virus. it's not binary fission like bacteria or other cells. And that you don't take one and break it in two and then 2 to 4 and so forth. It doesn't happen with viruses. You make the particles from parts. And that's reflected in these growth curves, which we will look at in more detail later on. We classify them by the Linnaean system, the binomial system, genus, species. Here's the typical hierarchical system. And we typically use genus and species for viruses. There are some families and some orders as well. And we don't usually go above the order in classifying viruses. We classify them by a variety of physical characteristics, the nature of the nucleic acid, the protein shell, whether there's an envelope or not, and how big the virus is. These all go into classifying the virus. But now in the most recent 5 years, genomics has really taken over in virus classification. So you can isolate a new virus. You don't even have to isolate a virus. You can get a sequence of a virus and look at it. You have your bioinformatics people look at the sequence, and they'll tell you where to classify it. You don't have to think about any of this other stuff. There's actually some controversy about this in the field, but genomics has become very important at classification. So here's an example. A research crew went to Antarctica, went down, I guess, to Antarctica. They drilled holes in the ice. There are apparently freshwater lakes down there, and they drilled holes in them, and they sampled the water, and they just sequenced the water. So they extracted nucleic acids from the water, and they sequenced it. It's called deep sequencing. And they found 10,000 new virus species just by sequencing it. You sequence it, and you search the database, and you say, there are 10,000 sequences here that are all different. I don't see any of them in the database. So you have new virus sequences. And it says here, in total, the viruses were from 12 different families, some of which may be completely new to science. So you can put a virus in a family just by looking at the sequence nowadays. So that's an example of what I was telling you. Here's an example of how you would classify a virus. There's a very nice site over in Switzerland called ViralZone that has all of this listed out. Typical taxonomy, we'll talk about classifying the viruses according to genome. Here's the family, the genus, and examples of different species. So family, genus, and species for classification. There is a committee that deals with the classification of viruses. It's called the ICTV, and they have a database which is hosted at Columbia. You can find every virus at this database. So far, there are about 40,000 virus isolates from bacteria, plants, and animals, placed in 3 orders, 73 families, 287 genera, and 1,950 species. But these are the ones that have been characterized. There are 1 million virions per milliliter of seawater, and we don't know what most of them are. So there's a lot more out there that we have to learn about. Now, we're going to now move on for the rest of today, and then next time talk about viral genomes. And the important breakthrough here occurred in the 1950s when it was shown that the viral nucleic acid genome carried the information needed to build viruses. Before that, we didn't know how new viruses or organisms were even built. Now this seems very obvious, but that was one of the building blocks of this field called molecular biology. You may remember that in 1944, a couple of investigators at the Rockefeller showed that the DNA of a bacterium was the genetic information, but for viruses that wasn't known until the 1950s. And that was done by these 2 investigators, Alfred Hershey and Margaret Chase, in 1952. They took a bacteriophage and they labeled it with 2 different kinds of radioactive isotopes, one to label the protein shell or one to label the DNA. And then they took these radioactively tagged bacteriophages, they infected bacteria, and they asked, where does the label go? So when they labeled the protein, they found that there was nothing left in the cell after the virus infected that cell. There was no radioactive protein. But if they labeled the DNA, you can do that with phosphorus, P32, they found that the label went inside the bacterium. So this was the first proof that the DNA of the bacteriophage went into the bacterial cell and started the infection. See, before this, we didn't know. We thought, well, maybe it's the protein. In fact, many scientists thought protein was the genetic information because you have 21 different amino acids and only 4 bases in DNA. So many scientists said DNA can't be genetic information. It's not complex enough. So this was a very important experiment to prove that. Now, next time we're going to go through all of the different kinds of viral genomes. But when we do that, I want you to remember that they all have to make mRNA that has to be read by ribosomes. All viruses on the planet follow that rule, no exception. And that'll help you sort it all out. It turns out that there are 7 different genome types among all the viruses. And David Baltimore, who was a Nobel laureate, used this insight to unify the classification of viruses. And he says, we'll put at the center mRNA, and then every virus genome has to get to it. So he said there's 7 types of viral genome. Actually, he said there were 6. Then we discovered a 7th later on. Double-stranded DNA, single-stranded DNA, single-stranded RNA of either plus or minus polarity, single-stranded RNA of plus or minus polarity, and then single-stranded plus RNA that goes through a DNA phase, and then double-stranded RNA. And then later on, a 7th class was added, double-stranded DNA with gaps. And we will talk about all of those in great detail. But this is a major unifying principle in virology. So we use this to classify viruses. And again, if you don't know this terminology, the plus strand is mRNA by definition. It has nothing to do with electricity, plus or minus. It's just the randomly assigned specification that means the strand that can be translated. So when I say plus RNA, I mean the strand that can be translated, and minus is the complement. In the strand of DNA, that's the plus strand, is the strand that would be the same as mRNA. So that's what we talk about when we say plus and minus strands and double-stranded RNA. So the Baltimore system allows you, knowing only the nature of the genome, to figure out all the steps that the virus has to do in order to replicate. So by the end of the next few lectures, you'll be able to look at any genome configuration and predict how it's going to replicate because of this Baltimore scheme. So it's a very powerful rubric that will allow us to get through those 7 different genome types. And here they are again: double-stranded DNA, gapped double-stranded DNA, single-stranded DNA, and then our RNA genomes, double-stranded RNA, plus or minus, single strand, and then finally our retroviruses, plus-stranded RNA with a DNA intermediate. So next time we're going to go through each of these and talk about how they work.